Hello, this is Mr. Merrier with Noteworthy Science. Today we're going to be doing subtopic B1.1 Carbohydrates and Lipids. Under the curriculum matrix, this falls under theme B, form and function, and under the level of organization of 1, molecules. Some guiding questions to keep in the back of your mind as you're learning this topic is 1, how does the variation in form allow for diversity in function? And we're going to be looking at this for both carbohydrates and lipids. And then again, looking at carbohydrates and lipids, we're really going to see how do they compare as energy storage molecules? In what way do they differ? How is each adapted to being their respected type of energy storage compound? This subtopic actually does not have any higher level learning outcomes. It is all standard level learning outcomes, and there are 13 of them. Let's begin. So to begin this subtopic, what we're really going to be exploring is the concept that you likely heard before, which is all life is carbon-based. And really, why is carbon the base of all the biomolecules that we're going to talk about? Things like carbohydrates, lipids, proteins, nucleic acids, all of these are carbon-based. So this is all really looking at the properties of carbon and what makes it such a versatile atom that allows it to make so many different structures and molecules. And I think before we go into that, let's do a very brief chemistry review. And let's consider what properties allow atoms to make more or less bonds. So recall that the outer shell of electrons are called the valence shell of electrons. And for atoms to be stable, they want to fill their outer shell. And this either means that they're going to acquire some electrons in order to fill a valence that is nearly full, or they're going to get rid of electrons in order to get their lower shell to be full. Now, if we look at a periodic table, the number of atoms in a period actually tells you what the maximum number of electrons are in each shell. So in the first period, there are 2 elements, meaning that there is a maximum 2 electrons in the first shell. In the second period, there are 8 elements, so that tells you that there is a maximum of 8 electrons in the second shell. And same thing with the third period, 8 elements across, 8 electrons total. So 2, 8, 8 in terms of the 3 shells. Now, if we look at carbon, for instance, carbon falls under group 14, or its valence is 4 electrons, meaning that it needs 4 more electrons. So that allows it to make 4 different bonds. And what carbon will do is it will form covalent bonds. So let's explore that. So carbon has 4 valence electrons, and it would be very unlikely for carbon to ever become ionic, i.e. to acquire 4 electrons or to offload 4 electrons. Rather, what it almost always does, especially in this course, is going to be forming covalent bonds. And recall, covalent bonds is just the sharing of a pair of electrons. And that force of attraction is between the positive nucleus and the negative electrons. So because carbon has 4 valence electrons, it could form 4 different covalent bonds. Now, what's quite amazing is the sheer variety of bonds that it can make. Carbon may share a single pair of electrons in a single covalent bond, which is represented by a single line. Carbon may also share 2 pairs of electrons in what's called a double bond, and represented by 2 solid lines. Or carbon may even share 3 pairs of electrons in what's called a triple bond. So if these are hydrocarbons, this would be an alkane, an alkene, and an alkyne. And so there's a huge amount of variety that carbon can already form. But that's not all. Carbon can also form a huge variety of molecular shapes. So carbon can form unbranched chains, such as in the case of many hydrocarbons. They can form branched chains, like things like in amino acids, or if a really good example would be looking at carbohydrates like polysaccharides. They can form linear chains, which means that the molecule does not display any kind of bending. They can form bent chains, something that we'll explore when we look at unsaturated fatty acids. And anytime that we have a cis double bond, you're going to get this bending nature, and so the molecule is no longer really linear, but it has this bent structure. And something that we'll explore a lot more deeply is the fact that they can actually form ringed structures. And not even just a single ring, but multiple rings. as is the case here of cholesterol, which is a 4-ring structure, or if we've already explored with things like purines that are a double-ringed nitrogenous bases. So there's a huge amount of variety that carbon can make. In addition, carbon-based molecules have really no defined length. They can be extremely short, like the shortest carbon molecule would be methane, which is a single carbon, but carbon can form enormous structures. I believe one of the largest structures I can possibly think of would be the protein Titin, which is composed of, I believe, over 30,000 amino acids. And carbon atoms alone, I believe, is over 170,000 carbon atoms. So we have enormous variety in the kind of structures that carbon molecules can make. And additionally, depending on what actually gets bonded to carbon molecules, called functional groups, they can actually alter the properties of that molecule. And so, for instance, some very common functional groups that we'll explore in biology would be things like an amino group that has an NH2 group coming off of it, a hydroxyl group, something that we've already explored a lot, especially with respect to polar molecules, is when there's an OH group attached to it. In fact, the functional groups here are things called alcohols. Or we have phosphate groups, something that we've explored already with respect to nucleic acids, but phosphates can be added to all sorts of molecules. We also have carboxyl groups or acids, where you have this double bonded oxygen and an OH group coming at the terminal end of a chain. A carbonyl group is when you have a double bonded oxygen in the middle of a chain. You can have a methyl group where you attach a CH3 group on top of a chain, and this is something that we'll explore especially when we look at epigenetics, where we can actually modify histones by adding methyl groups to it, a process called methylation. Or you can even get sulfhydryl groups, where we actually add a sulfur group to a chain. Carbon does not just form bonds with itself, it can form bonds with a number of different molecules. So for instance, it regularly bonds with oxygen, hydrogen, nitrogen, phosphorus, and sulfur, as in the case of the 4 major biomolecules. Now, as a quick aside in terms of nomenclature here, organic molecules refer to any molecules that have carbon and hydrogen bonded covalently together. So, for instance, ethanol would be considered an organic molecule. Even though it contains oxygen, it contains carbon and hydrogen bonded covalently. Same thing with methane. This would also be considered organic. However, something like carbon dioxide or ammonia, even though both of these molecules are highly associated with biological systems, they would not be considered organic because they do not have carbon and hydrogen bonded covalently within their structures. Yet more variety when we consider carbon molecules is the fact that you can have even the same exact chemical formula, but slightly different structures, what are called isomers. Or a special kind of isomers that you'll look at to maybe if you explore IB chemistry are what are called enantiomers. And these are effectively mirror image isomers of each other. So these may look extremely similar to each other, but from a biological point of view, these have very different interactions with enzymes and with metabolic pathways. So if you ever see the names of certain structures, you may see the letter S or R or D and L in front of it, which designates the right-handed or left-handed version of that molecule. So this is yet even more variety that carbon molecules can make. So the overall summary here is that life is carbon-based, likely because of the sheer variety that carbon allows. Carbon can form single bonds, double bonds, triple bonds. It can form bonds with itself and a number of other types of atoms. It can form a huge variety of structures in terms of bent or linear or branched or unbranched or ringed or multi-ringed. And it can even have multiple functional groups, and it has no real minimum or maximum length. It has huge amounts of variety that it can form. In fact, you can form more different kinds of molecules with carbon than any other combination combined that we know of. So life's sheer variety is really all to do with carbon's sheer variety. So the nature of science learning outcome here is exploring the concept that science is an international and collaborative endeavor. And a reason that this is possible is because we have a lot of uniform conventions that are independent of language. And one of those conventions is our agreed upon system of measurement, something that we call SI units, or the International System of Units. If you're wondering, why is it SI when it's the International System of Units? This comes from the French, which is Système International d'Unité. But really, what we're talking about here is that these units are used across the globe, with maybe an exception of the United States. But just about every single scientific endeavor are going to use the same kind of units. And these units can broadly be classified into two kinds of categories, that being fundamental units, and these would be variables that are not derived from any other units, meters or seconds or kilograms or Kelvin or amps. These are all fundamental units. But then there's also even derived units, things like degrees Celsius or meters per second or watts or volts, which are derived from other kinds of units. So the idea here is that it doesn't matter what language I'm doing science in, I'm going to be using the same kind of units, and this is going to allow international collaboration and international understanding as to what I'm really doing. Now, a fundamental skill in science is being able to convert between different units. And the good news about the International System of Units and metric, really, they're all base 10, which makes things very, very easy to convert. So for instance, what I have here is just kind of an outline as to the major kind of units that we'll explore in biology. So here would be what the base 10 value would be for this particular unit. This would be the prefix that would be used to describe this unit, and this is the symbol that we use when expressing this unit. So as you notice, they're all base 10, and with only one exception of centimeter, they're all base 1,000. So it makes conversion very, very easy. Now you might be wondering, is this just making things more complicated? Why have all these extra prefixes? Can't we just work in a base unit like meters or seconds? Well, the issue with that, as I'm sure you can probably already understand, is that the prefixes are used to express a unit more practically. For instance, I could describe myself, my height in meters, and that might make sense. But if I want to express the size of the diameter of a nucleus, expressing that value in meters would become an extremely small value, 0.000000, etc., where the value would become so small. Or vice versa, if I want to express how far away a galaxy is, if I'm only going to use meters, my number will become enormous. So rather, we can use prefixes that kind of make sense for the context. So let's practice actually doing a conversion. Convert 0.025 kilometers into micrometers. So let me show you how I would approach this type of problem. I want to convert kilometers to micrometers. Now, kilometers is 10 to the 3, and micrometers is 10 to the minus 6. So what is the difference between these two? 10 to the 3 minus 6 equals 10 to the 9. So there's actually a billionth of a meter difference between these two. So I'm going to use dimensional analysis in order to really make sure that I understand my conversion here. So I'm going to divide by kilometers. So we know that there's a 10 to the 9 difference between the two. So you have to kind of ask yourself the question, should my number be getting larger or smaller? Well, if I'm going from kilometers to micrometers, you can kind of ask yourself the question, is there a bunch of kilometers in a micrometer or a bunch of micrometers in a kilometer? Well, there's a lot of micrometers in a kilometer. So there is 10 to the 9 micrometers in 1 kilometer. And so when we do this conversion, kilometers cancel out and you're left with this. So on your calculator, what I would enter is 0.025 multiplied by 10 to the 9, and I'm going to get a pretty large number. So This number is fairly large, and trying to read this number quickly is a little cumbersome. So what we use a lot in science is scientific notation. Now, luckily, your calculator can actually convert for you. What you're going to hit is you're going to hit mode, and you're going to hit science, and then hit second function mode to quit, and hit enter, and this will convert this number for you. So this also equals 2.5 times 10 to the 7 micrometers. Before we go into the structure of carbohydrates and lipids specifically, we first want to make sure that we kind of understand just how biomolecules are built and how they're broken down. Now, I've alluded to this briefly when we looked at nucleic acids and how a phosphate sugar backbone was actually built using condensation reactions and how they're broken using hydrolysis. But I think now we're going to formally look at this. So firstly, every time that I'm mentioning biomolecules, what I mean are the 4 major classes, and that being carbohydrates, proteins, nucleic acids, and lipids. Now, the name biomolecule, bios means life, so life molecule. Well, what does that mean? Well, these molecules aren't literally alive, but they're molecules that are crucial and found in all living systems. So all of these biomolecules exist as simple monomers, and they can be combined together to be more complex polymers. So the interaction that we're going to be looking at a lot is how do we go from monomers to polymers and polymers to monomers? Well, again, we've already alluded to this, but any time that we are combining monomers together to build something larger, this is anabolic. And anytime that we're breaking something down into something smaller and simpler, that is catabolic. So a condensation reaction are how all biomolecules are going to be built anabolically, meaning that we're going to take 2 smaller, simpler molecules and combine them together to form a larger, more complex molecule, or monomers to polymers. And in doing so, every single time a new bond is going to be built between 2 monomers, that's going to result in a water molecule as a byproduct. Hence the name condensation reaction. This also goes by another name, which is dehydration reactions, meaning the removal of water. But the course uses the term condensation, so I'm going to continue to use this term. And so all of the biomolecules that we look at, keep note of this pattern. One monomer is going to have at least an OH group, and the other one is likely going to have an OH group. And then what ends up happening is that an H and an OH come out to form water, and then a covalent bond is made linking both monomers together. So in this case, this would be a monomer and a monomer forming a dimer and a water molecule. So for every new bond being made, there's going to be a water molecule being made as a byproduct. Now, these types of reactions, as mentioned, are anabolic, and overall, in terms of energy levels, it would mean that these overall require energy. We'll explore the concept more in terms of energy required and energy released when we look into metabolic processes in C1.1. Now, the converse of this type of reaction would be hydrolysis reaction. And these reactions are where you're taking a larger, more complex polymer and breaking it down into smaller, simpler monomers. And to do this, we are using a water molecule to break the bond between these two things. Hence the name hydrolysis, hydro water, lysis split. So it literally means splitting with water. So again, if we were to look at a covalent bond, in this case a covalent bond between 2 monomers, this water molecule is going to be used as a reactant in order to break apart this bond. And in so doing, you're going to end up with a hydroxyl group on one monomer and then an extra hydrogen on the other. And this is where that water molecule was derived from. These types of reactions are catabolic in nature and overall would release energy or be exothermic to the environment. So now let's explore how condensation and hydrolysis reactions can be used to both build and break down different biomolecules. And we're going to look at this process in all 4 biomolecules: carbohydrates, nucleic acids, proteins, and lipids. With nucleic acids, recall that their monomers are nucleotides. If I build 2 nucleotides together, I can form a dinucleotide. And if I form many nucleotides together, then I can form a polynucleotide, or specifically DNA if it's made up of deoxyribose, or RNA if it's made up of ribose. And so the way in which a phosphate sugar backbone is formed is through condensation reactions between the hydroxyl group of this 3' carbon and the hydroxyl group of the phosphate of an adjacent nucleotide. So these would actually get removed, forming water as a byproduct. And the resulting bond formed would be a phosphodiester bond. Recall phosphate, and then I have a di-1-ester, 2-ester right here. And so the reverse process can also occur. If I take this water molecule, I can use it to break this bond and actually break apart into 2 separate nucleotides. Now, relevant enzymes that undergo these processes, an anabolic nucleotide enzyme would be DNA polymerase, an enzyme that builds DNA nucleotides together during DNA replication, or RNA polymerase that's used during transcription to build a new mRNA, or even DNA ligase, which is also used in DNA replication in order to join different nucleotides together. Some catabolic enzymes that break down nucleotides would be, for instance, endonucleases. And these actually will sever the phosphate sugar backbone, and they can even be used in bioengineering processes where things like PCR, where we can actually break down certain DNA strands at particular sequences of our choosing. Carbohydrates monomers are called monosaccharides, meaning single sugar. If I add 2 monosaccharides together, I can form a disaccharide, 2 sugars. And if I take many of these together, I form a polysaccharide, or many sugars. And all of these are all combined using what's called a glycosidic bond. So recall that carbohydrates have plentiful amount of hydroxyl groups, and by removing an OH and an H from 2 adjacent carbohydrates or monosaccharides, I can create water as a byproduct, and similarly I can use water in hydrolysis to break this glycosidic bond, reforming these individual monosaccharides. Now there are plenty of examples of this, and each single kind of combination is catalyzed by a different enzyme. If I'm combining 2 glucose molecules together, I can use maltose synthase to create maltose. If I'm joining a glucose and a galactose together, I can use lactose synthase in order to produce lactose. If I combine glucose and fructose together, I would use sucrose synthase in order to form sucrose. Now, we also have enzymes that can break all of these disaccharides down. Maltose, maltase, lactose, lactase, and sucrose, sucrase. So hopefully you're starting to see some kind of a nomenclature pattern here. The suffix ase is used to designate an enzyme. Synthases are usually anabolic enzymes, and catabolic enzymes are usually named after their substrate. So maltose is maltase because maltose is the substrate. Now, in terms of polysaccharides, if I take a bunch of alpha glucose together, I can form either glycogen or amylose or amylopectin or starch. And if I take many beta glucose molecules together, I can form something like cellulose. Now, proteins, something that we'll look at more specifically in B1.2 in the protein subtopic, but this will be a good preview. The monomers of proteins are amino acids. If I join 2 amino acids together, I get what's called a dipeptide. And if I take a bunch of amino acids together, many, 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 I get a polypeptide, or really colloquially a protein. So what exactly is going on here? Well, peptide is named after the bond that's formed. In this case, a peptide bond. So let's explore this. If I have one amino acid and another amino acid, then the OH group from the carboxyl end is going to combine with the hydrogen on the amino end. These are going to form a water molecule, and then what's left is that the carbonyl carbon and the nitrogen are going to form a covalent bond. That covalent bond, what's called a peptide bond. So these peptides are always going to form in the same way. Nitrogen, carbon, carbon, nitrogen, carbon, carbon. So in fact, I always tell myself this when I'm drawing a polypeptide, NCC, 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 because this is the way in which they're going to form. Amino acids are the most variable of all biomolecules, and that's specifically because of this R section here. What exactly does R mean? Well, R refers to residual or side chain group, and there's actually 20 different things that this R can be. So this is just a generalized amino acid. And we'll explore this concept more in HL when we look into proteins. Now, a really cool example here is that the enzyme that builds all of the proteins in our bodies is actually not protein-based. It's actually rRNA, or ribozymes. These are found within ribosomes, and these are actually what are catalyzing the peptide bonds between amino acids. Now, catabolic enzymes that break down proteins would be things like peptidases. Again, the substrate would be a peptide, and ase for an enzyme. Or we have specific kinds of enzymes located in different parts of the body. In humans, within the stomach, we have a catabolic protein enzyme called pepsin. And what's secreted in our duodenum by the pancreas is another kind of peptidase, which is called trypsin. Now, finally, will be lipids. Now, lipids are slightly complex in the sense that, unlike the other three biomolecules, the polymers are not made up of repeating monomers. They're actually made up of several different components. So triglycerides are actually composed of one glycerol molecule and 3 fatty acid molecules. And so the hydroxyl groups of the carboxylic acid of each fatty acid is going to combine with the hydrogen of the glycerol molecule, and the resulting bond will be ester bonds. So that means in order to build a single triglyceride, 3 water molecules would be formed as a result. And vice versa, if I were to fully break down a triglyceride into 3 fatty acids and a glycerol molecule, I would require 3 molecules of water to break each of these ester bonds that are formed. Now, triglycerides are not the only polymer that we'll explore here. Another one is of very importance in biology, and that is a phospholipid. So the name here comes from the fact that it is composed of a phosphate and a lipid. So we still use the same glycerol molecule, and this time we're going to bind it to 2 fatty acids and 1 phosphate group. But again, notice the trends here. Hydroxyl group, hydroxyl group, hydroxyl group, and once again, 3 water molecules would be formed as a byproduct, and 3 different water molecules would be used to break a full phospholipid down. I don't think technically we can call lipids polymers because they're not made up of repeating units, but note that they are composed of different kinds of monomers. Because phospholipids are typically drawn in this simplified form, a lot of students mistakenly think that phospholipids are only composed of a phosphate and fatty acids. They forget about this very important glycerol here. So don't forget that a glycerol molecule is also used in a phospholipid. So overall, even though these 4 different biomolecules are quite variable in their structure, they all have a similar pattern when they do anabolic and catabolic reactions. They will all undergo very similar condensation and hydrolysis reactions. What we're going to be going into now is the guiding question, or the overall thing that we are going to look at, was how does the variety of these structures allow for all of the different functions that they do. So we're going to look at this in this subtopic really in detail with carbohydrates and with lipids. So for carbohydrates, let's first of all, what exactly are carbohydrates? Well, luckily it's kind of in the name. Carbo, carbon, hydrate, water. So they're composed of carbon, hydrogen, and oxygen. And specifically, if you're wondering, well, aren't every biomolecule made up of carbon, hydrogen, and oxygen? Yes, but the proportion of oxygen and hydrogen or oxygen rather, is very high compared to carbon, right? There's pretty much almost always a 1-to-1 ratio between oxygen and carbon. So that's really significant because that means that there's, as we already know, a ton of hydroxyl groups. And if there's a lot of hydroxyl groups, then that implies polarity. So this is why most carbohydrates are very polar, because of the huge abundance of hydroxyl group all around them. So they're monomers, as we've said. All biomolecules can have monomers that can combine into polymers and break down into monomers, right? So the monomers for carbohydrates are called monosaccharides. Basically, it just means one sugar. So monosaccharides, this is going to be the building blocks of carbohydrates. And we've already explored some of these when we looked at nucleic acids, right? So just as an aside, this is not necessarily a learning outcome, but if you were to Google image search or you were to look up some carbohydrates, you may see even glucose in this weird linear form. But a lot of these carbohydrates exist both as ring structures and as linear structures. We're mainly going to deal with ring structures in this class. Let's look at two different carbohydrates that we've already explored when we looked at nucleic acids. We should be well familiar with these by now. Both of these are pentose sugars, and again, pentose not with respect to a pentagon, but with respect to 5 carbons, right? 1, 2, 3, 4, 5. And the two major pentose sugars that we've explored were that of deoxyribose and ribose. The only difference being is that ribose had one extra oxygen and therefore an extra hydroxyl group than the deoxyribose did. Now the other monosaccharides are what I would call isomers. So as a review, an isomer is any chemical that has the same chemical formula, so that means it has the same amount of each atom, but they are arranged differently. So in these cases, a lot of the monosaccharides are actually isomers of each other. So let's explore some of these isomers. And a lot of them you've heard of before, that being glucose, galactose, and fructose. Now note that all of these are C6H12O6. They all have the same number and type of atoms, but they are arranged slightly differently. I want to pay special note to these two right here. Both of these are glucose. However, there's a very, very small difference between them, and that is the actual orientation of the hydroxyl group on carbon 1. In alpha glucose, this hydroxyl group is in the downwards direction versus beta glucose, it is in the upwards direction. Now, you might be wondering, who cares? Does this actually matter? Absolutely it will, and we'll explore why it does when we explore polysaccharides, as in how do these combine together. Some polysaccharides are made up of alpha, but others are made up of beta. And these are not the same biologically. So we'll explore why this very small difference actually matters in terms of structure. And other monosaccharides would be galactose and fructose. I can have all kinds of combinations here. Probably the monosaccharide that you've heard of the most, and we will absolutely be discussing the most in this course, is glucose. Glucose has enormous applicability in biological systems. So there's a specific learning outcome that we explore. What properties does it have that make it such a good energy source? Because this is going to be the main metabolic substrate for chemical reactions. It's going to be glucose. So what about glucose? What properties does it have? So for one, as we've already discussed, the high abundance of hydroxyl groups here make it polar and thus very soluble in water. And that's relevant because it means that it has very good transportability. So it means that it's going to be, one, able to dissolve within blood plasma, it's going to be able to dissolve within cytoplasm, which is mainly water, and it's going to be able to dissolve within the interstitial fluid, or the fluid in between cells and tissues. But this also means that we can transport it very, very easily within the blood, because the blood is mainly plasma. So it means I can move glucose from one part of the body to the other part of the body very, very easily. In addition, if I want an energy source that I can move around easily, I don't really want that energy source to be unstable, meaning that it reacts like very, very quickly. I want it to be relatively stable. And it is, because it is made up of fairly stable covalent bonds. And so glucose isn't going to spontaneously break down or react typically. It's going to require enzymes in order to do any of the reactions that we do. And then finally, well, if we're going to use something as an energy source, it better have a pretty good energy yield, and it does. So when we undergo cellular respiration, both aerobic and anaerobic, we can use glucose as a substrate, and that releases actually a fair amount of energy per gram. So it's actually a fairly good energy source, and that's why it's used by all animals in terms of energy. Same, and not just animals, plants as well. So these are how monosaccharides are built, but as we've discussed, we can join a bunch of these monosaccharides together to make polysaccharides. If one carbohydrate was called a monosaccharide, two carbohydrates joined was called a disaccharide. Many carbohydrates joined are polysaccharides. And in this course, we're going to explore 4 different polysaccharides, that being glycogen, amylose, amylopectin, and cellulose. So let's start firstly with glycogen. So glycogen, as a polysaccharide, is entirely composed of alpha glucose, and its structure is highly, highly branched. Now, what does that mean? So if we zoom in to glycogen here, here is my main chain. And notice, if I'm numbering the carbons, and we number in the same way that we numbered the deoxyribose and ribose, right? Clockwise, starting from the 1 carbon below the oxygen. So that would be 1, 2, 3, 4, 5, 6. If this carbon is glycosidically bonded to this carbon, then we can call this bond an alpha, because it's between alpha glucose, 1,4 glycosidic bond. So with this sounds kind of fancy, but it's actually providing you a lot of information. Alpha, in that the 2 monomers are alpha glucose. 1,4 designates which carbon in the ring is actually being bonded together. So that is 1,4. So this bond in the main chain is alpha 1,4 glycosidic, alpha 1,4 glycosidic. And you get that over and over and over again. So if you look at this structure right here, all of these would be 1,4, 1,4, 1,4, 1,4, 1,4, 1,4, 1,4, 1,4, etc., right? But notice that off of this chain, there's a bunch of other chains coming off of it. That's where the term branched comes from. So how is that actually happening? Well, if we look back here, the 6th carbon can also bind to the 1st carbon of another glucose. So unsurprisingly, we can name this alpha 1,6 glycosidic bonds. So any alpha 1,6 bonds are the ones coming off of that main chain, if that makes sense. So anywhere where we get a new branch, there's going to be an alpha 1,6 bond coming off, starting a brand new branch, and then all of these would be 1,4s again. So that 1,6 is effectively where a new branch begins. So the more 1,6 bonds there are in a molecule, the more branched it is, if that makes sense. Here is a molecule of glycogen. It's helical. In fact, you can look straight down this way, and that's all of this would be 1,4, 1,4, 1,4, 1,4. Look at this molecule and deduce how many alpha 1,6 bonds there are in this molecule. 2. And how do we know that? There's two branches coming off. Anytime there's a new branch, that's a new 1,6 bond happening. Now obviously this is a very small portion of the glycogen molecule. Glycogen is highly branched. This is just one section of it. Now if you take a look at this molecule, you may be wondering, this looks really similar to glycogen, and you'd be right. They have a lot of similarities. They're also, similarly to glycogen, they are entirely composed of alpha glucose. Similarly to glycogen, they are also branched. However, if you, and I tried to show these two in this drawing here, but if you were to compare the relative proportion of alpha 1,6 glycosidic bonds, they're far more prevalent in glycogen than they are in amylopectin. So while both glycogen and amylopectin are branched, on average, there'd be significantly more branching in glycogen than in amylopectin. So again, if you were to examine the structure of amylopectin, you would likely see something incredibly similar. In this one, they just showed one branch, so there'd only be one alpha 1,6 bond here. But again, keeping in mind that this is only a small section of it. Now, you won't be responsible for, let me give you a diagram, and be like, which is it, glycogen or amylopectin? You don't, no. You wouldn't be, you would just basically understand that glycogen is significantly more branched than amylopectin is. So amylose is also, yet again, entirely composed of alpha glucose, but it is unbranched. That means that its entire structure is only going to be alpha 1,4 glycosidic bonds. There's a complete absence of alpha 1,6 glycosidic bonds. So if we explore amylose, as you can probably already imagine, you'll notice that there is a complete lack of alpha 1,6 glycosidic bonds, only alpha 1,4 glycosidic bonds. And just like before, that main chain forms this helix, or this helical structure with all of them together. What do all of these mean together? Well, now that we understand the structural differences and similarities between glycogen, amylopectin, and amylose, let's now use that guiding question, or overall, how is their structure relating to their function as energy storage molecules? So all of these molecules are used as a short-term energy storage in organisms. So organisms like us and plants, they're not going to store a bunch of glucose as individual monomers. They're going to store them as polysaccharides. Glycogen is the polysaccharide that animals like us use. So all of our glucose is stored as a polysaccharide as glycogen. Now, glycogen can be found in all kinds of different tissues, but the two primary organs that store the most glycogen are your liver, and what other organ would you say have very high energy demand and would make sense for them to have a lot of glycogen? Skeletal muscles. Skeletal muscles also store glycogen so that they have ready access to breaking that down and then using it to generate ATP. Believe it or not, fungi also use glycogen as their energy storage, but plants do not. They store it as I'm sure a term that you've heard before, that being starch. So what exactly is starch? Well, starch is a combination of both amylopectin and amylose. In fact, if you looked at a typical starch thing, then that proportion would be mainly amylopectin and also some amylose. So starch is really a mixture of these two different kinds of polysaccharides. This is how plants store their glucose. So when plants photosynthesize and generate glucose, they will then store that glucose as either amylopectin or amylose. So when we eat plants, we're eating starch, we're eating really glucose. And it's the same exact monomer that we can then use to build glycogen, because they have the same building block, so to speak. What makes glycogen and starch such good molecules for short-term energy storage? Well, for one, starch and glycogen, because it's branched, it can actually be compacted. And that means that I can store quite a lot of it in a small area. So where we store it would be in the liver and in muscles, but in plants, they could store this either in plastids, called like chloroplasts or amyloplasts, or anyone know a specific storage organ in some plants? We eat them. Called tubers or potatoes, right? What exactly is a potato? It's a giant thing of starch, right? So if plants want to store a lot of starch in a singular area, that's like a tuber, that's like a potato. And that's why they're so high in starch if you eat potatoes, right? So the idea is that by being branched, they can actually compact quite well, and you can store quite a lot of it in an area. Something that we haven't quite explored yet is the concept of osmosis. This will be a really big recurring theme in biology. We'll be coming back to osmosis a lot in this course, but as a preview, let's say I have a beaker, let's say I have a partially permeable membrane here that does not allow the flow of solutes, but it does allow the flow of water. What direction will water flow? Water will flow from areas of high water potential or low solute concentration to an area of high solute concentration or low water potential. Now, this is significant because if a cell just stored a bunch of glucose, then it would actually have a fairly high solute concentration. And if that's true, if I have a cell and I'm going to store a bunch of glucose inside of it, then what will happen to this cell? Water will flow in, it will become turgid, but we don't have cell walls. So what's going to happen to us? It'll lyse, it'll burst open. So the problem is that if we continually store glucose within, it will actually contribute to osmotic potential, meaning that it's going to take on a bunch of water. That is not very efficient. So instead, if we store it as a polysaccharide, this does not contribute to osmotic potential. So it will not influence osmosis. So I can store polysaccharides without the risk, so to speak, of gaining a bunch of water if I want to store something inside of it. Is there an advantage to being branched versus unbranched? Yes, there's a big advantage. And that has to do with a few things. One, it's going to alter the relative solubility of these molecules. Now, generally speaking, glycogen and starch have actually quite low solubility in water. And that's because even though these are polar molecules, they're really large. They may be able to dissolve in water, yes, but you're going to hit saturation point at a fairly low concentration. However, a branched molecule will actually have a higher solubility than an unbranched molecule. Can anyone deduce why that might be? Compare these two molecules. Why might this molecule have a higher solubility in water than this molecule? So generally speaking, if you were to compare the relative solubility of a branched versus an unbranched structure like this, the branched carbohydrate should have more solubility than an unbranched one. So if you were to compare the relative solubilities of like glycogen, amylopectin, and amylose, then in that order is what you would predict their relative solubility. Glycogen should have the highest solubility, and amylose should have the lowest solubility. So remember, solubility has to do with this interaction with water. So by having, think about like the little space that water has to be. There's more areas for it to form hydrogen bonds than something like this. There's just more space, if that makes sense. And then lastly, what's another advantage to being branched? Well, recall that I said carbohydrates are a very good short-term energy storage molecule. That means that this is a molecule that I can very easily build into a polysaccharide, and I can very easily break. So why does the branch structure matter? Well, remember, the only way that we're going to be able to either hydrolyze or do condensation reactions are enzymes. That means anywhere where enzymes are free to then use in their active site to catalyze that reaction, that's where it's going to occur. Look at the physical difference again in space between a branched versus an unbranched structure. By having a branched structure, that means that an enzyme can really attach here or here or here or here or here or here or here. There are so many different possible active sites for enzymes to either break pieces off or to add pieces on. It's very easy. I can build off of here, I can build off of here, I can build here, 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 here. If you compare this to an unbranched structure, then where can the enzymes operate? Effectively at the terminal ends. They can build off of here or break off of here. So it still works, but the branch structure is highly favored because it's much, much easier to add subunits on and break subunits off. So the branch structure is actually quite useful in its function as a short-term energy storage molecule. Now I mentioned that we're going to be studying 4 polysaccharides. So far we've only looked at 3. Well, the 4th polysaccharide has nothing to do with energy storage. Instead, it has everything to do with structure, and that is cellulose. So cellulose, where its name kind of comes from, is it's found in the cell walls of plants specifically. So it's going to have a lot of differences now between cellulose and the other 3 polysaccharides. It is composed of beta glucose, not alpha glucose. So all of the monomers that make up cellulose are beta glucose, and look at the relative orientation of the glycosidic bonds here. The monomers are actually 180 degrees flipped from their neighbors. So in this up, down, up, down, up, down pattern. So you have beta 1,4 glycosidic bond, beta 1,4 glycosidic bond, beta 1,4 glycosidic bond. Also notice that there are no branches. Here is cellulose. What differences do you see between cellulose and the other 3 polysaccharides? It's not helical, it's linear, or like as in these sheets. It's also unbranched. Now, why is this going to be relevant? Cellulose is actually going to be multiple sheets stacked on top of each other. If you add a bunch of these sheets in proximity to each other, well, we have a bunch of polar groups near each other. What happens when we place polar groups near each other? Hydrogen bonding. So if I stack almost a bunch of cellulose sheets, one on top of the next, you form a lot of these hydrogen bonds between them, sometimes referred to as cross bridges between one cellulose sheet and another cellulose sheet. It's actually almost similar to, if anyone here is familiar with fiberglass, or if anyone here is familiar with maybe paper mache, where the more layers that you add, the stronger and stronger and stronger the structure becomes. Where if anyone knows how fiberglass or paper mache works, is you take some kind of material like a mesh, and then you embed it in glue, and then you stack these one on top of the next. So the structural component is the actual polysaccharide, but where its strength comes from is the interaction between the polysaccharides. That's what gives it its strength. It's really the hydrogen bonds that form from one sheet to the next. So cellulose actually has tremendously high tensile strength. So it means it can resist forces very, very, very well. And in fact, take a look at how this structure is actually found. If you take a cell wall, you can actually they're arranged in fibers, and then each of these fibers are composed of many microfibrils, and each of these are these polysaccharide lines. What kind of structure does this remind you of? A wire or a cable? If you take a look at what a cable looks like or what a wire looks like, it's just a bunch of wires or materials all interlaced with each other, causing really high tensile strength. I mean, we use these for cranes, we use these for bridges. They have enormous amount of strength. And the same thing is true here. This is a big biological significance between beta and alpha glucose. Our enzymes cannot break down beta glucose chains. We have enzymes that can sever alpha 1,4 and alpha 1,6 glycosidic bonds, stuff like amylase. Amylase is an enzyme that can break down starch, for instance, but these enzymes do not operate on beta 1,4 glycosidic bonds. So we lack the necessary enzymes to break this down, which means that when we eat materials that are high in cellulose, like a lot of plant materials, that effectively moves through our digestive tract relatively intact. Now you might be like, well, that seems like a big waste of time. I'm not getting any nutritional value out of fiber, but it's actually allowing you to be regular, so to speak. It prevents constipation. So getting fiber in your diet is actually a really important thing to have regular bowel movements in that sense. The reason that other mammals, for instance, things like cows or sheep, the reason that they can process cellulose is because they actually have microorganisms in their gut that are breaking that down for them. The summary of this page really has to do with what are the building blocks of carbohydrates, what properties do monosaccharides, specifically glucose, have as a really good short-term energy molecule, and what are the 4 major classes of polysaccharides, and how do their properties help inform their ultimate function in different organisms. The same way in which we just studied carbohydrates in terms of what are the building blocks, what are the polymers, and how do these structures help inform the properties, we're going to do the exact same thing now, but with lipids. Firstly, something that we've already started to explore, a fundamental property that we care a lot about for lipids is that lipids are hydrophobic, meaning that they do not make interactions with water. So in aqueous solutions, lipids are insoluble. So the question is, why? Well, because lipids mainly are composed of non-polar covalent bonds. Specifically, a lot of lipids have long hydrocarbon chains, like this. And a hydrocarbon is just something that is composed of only carbon and hydrogen. And if you look at a hydrocarbon chain like this, there is no real partial charges being formed here. So as a result, there's really nothing for water to form hydrogen bonds with, which is why when you place lipids inside of water, they make separate layers. They will not interact with each other. Even if you try to mix it around, they will ultimately reform those 2 layers. They won't interact. This is not to say that you can't dissolve lipids. You can, just not using water as a solvent. You would have to use a different solvent. And sometimes these are referred to as organic solvents. And we'll actually use some of these solvents later when we do chromatography, where we try to dissolve different pigments within leaves. You're not going to be able to dissolve those with water. We're going to dissolve those using organic solvents. Some organic solvents that would be able to dissolve lipids would be something like propanone or toluene or cyclohexane, or different kinds of solvents that are able to interact with lipids, just not water. So, what exactly are lipids composed of? Well, the answer to this question is slightly more complex than it was for nucleic acids and carbohydrates, because all nucleic acids were composed of nucleotides. All carbohydrates were composed of monosaccharides. With lipids, it's slightly more complex because there's more variety. So I will say that just about all lipids have at least some fatty acids. So a fatty acid is effectively, it's a carboxylic acid group, right? We have a carboxyl group, and then we have a hydrocarbon chain. These fatty acid groups are a major component of a lot of different lipids. However, another exception here is that a polysaccharide were just a bunch of monosaccharides combined. A polynucleotide were just a bunch of nucleotides combined. A protein was just a bunch of amino acids combined. Again, lipids aren't quite that simple because they're actually composed of 2 or more different components. We build these into triglycerides, meaning that we're combining three of these with one glycerol molecule. Now, where lipids vary is in this chain. So there are three different major varieties that we're going to look at. The first one, and probably the simplest example, would be something called a saturated fatty acid. Now, saturated in this case means that every single carbon-carbon bond is single. So single bond, single bond, single bond, etc. So the term saturated comes from the fact of there's like the max number of hydrogens possible. Because all of these are single bonds, it's saturated with hydrogen. It's where that name comes from. So a saturated fatty acid has all single carbon bonds. This is often abbreviated as a skeleton drawing like this. That's what this implies. This implies all single bonds between the carbons. If you ever see a structure that looks like this, it's implying that this is a saturated fatty acid. What are the uses of it and where do we find it? So they are used as long-term energy storage molecules in specifically endothermic animals. And these are animals that you may know as warm-blooded. So these are animals that physiologically maintain a constant body temperature. So mammals are endothermic, and so are birds. Birds are also endothermic animals, meaning that they maintain a stable core body temperature. If my body temperature ever rose above, let's say 36, 37 degrees, my body would automatically make physiological changes to drop my temperature. If my temperature was below that threshold, my body again would make automatic changes to raise my body temperature. We'll explore this when we study homeostasis. Now, where do we find these? Well, unsurprisingly, we find them in meat of endothermic animals, right? So things like beef, pork, poultry, all of these types of animal products all are going to be rich in saturated fats. So anything that's really derived from these animals, any fats derived from these animals are going to be saturated fats. And specifically, they'll be stored as a triglyceride. So a triglyceride entirely composed of saturated fats would look something like this. Here's my glycerol, and here is three different saturated fatty acids. This is what we would term fat. Fat, in this case, is solid at room temperature. And this should not be really surprising. If you've seen steaks, if you've seen chicken, any fat that you see, well, it's solid. You can literally, it's usually slightly rubbery or slightly chewy. You can see it's usually whitish or yellowish in coloration. It's solid at room temperature. Well, why is that? Well, keep in mind that if I want to change different phases of matter, let's say from a liquid to a gas, all that means is that I'm providing more energy and thus breaking intermolecular forces that allow it to separate as a gas, right? Or vice versa, in back, less energy, more interactions, it'll be a liquid. Now in this case, if something is solid at room temperature, then that implies that it has a fairly high melting point. So the melting point of a lot of saturated fats are above room temperature, thus they remain as solid. Now why is that? Well, because all of the chains are saturated, it means that they can pack together really tightly. And as a result, they have a lot of strong, regular intermolecular forces. Now, we're not going to explore a lot of those forces in biology. If you study chemistry, you will likely study some of these. So in this case, because they pack really, really well, it's going to take a lot more energy for this to change phases of, or states of matter in this case. So this is why they are at a higher melting point and why they tend to be solid at room temperature. Now, what about if my fatty acid has a double bond? Well, now it matters the relative position of the hydrogens. Remember I said before that when double bonds are formed in a structure, you can start to get bending properties in a molecule. Well, this is how this happens. This would be termed a monounsaturated cis fatty acid. What does that mean? So mono, unsaturated, mono, 1. Unsaturated means that now there's actually 2 less hydrogens than there was before. And mono because there's only a single double bond. So this would be a mono unsaturated. Now cis means same, right? You've likely seen this with respect to gender identity. So if someone is a cis male versus a trans male, so I would be a cis male, meaning that the gender assigned to me at birth is the same as what I identify with now. Same, right? Trans would be different than what was assigned. So in this case, cis fatty acid means that the 2 hydrogen groups are on the same side. And in this case, that causes a bending nature to occur in the molecule. Because now there's an uneven structure, and anywhere where there's going to be a cis double bond, that's going to cause a bend in the chain. Can you get more than 1 double bond? Yes. And you can probably already deduce what the name of this is going to be. A polyunsaturated cis fatty acid. And now you can get many here. You can get 2, 3, 4 double bonds. In fact, you probably know of these already if you've heard of things like omega-3 fatty acid. But a polyunsaturated cis fatty acid now I have multiple double bonds, and each one of these is going to cause a kink or a bend in the chain. So where do we find these in nature? Just like in endothermic animals, these are also used as long-term energy storage, but they're not so much found in endotherms, they're found in ectotherms, or what you would know maybe as cold-blooded animals or cold-blooded organisms. So things like a lot of fish or in plants. Plants primarily store their lipids as unsaturated fatty acids. And again, you've likely have these in your diet, maybe without realizing, and that would be things like vegetable oils, olive oil or canola oil, sunflower oil, soya oil. All of these are plant-derived, right? Or you can have things like legumes, things like almonds, or things like walnuts, peanuts, sesame seeds, and primarily a lot of fish. So things like salmon, tuna, sardines would be very rich in polyunsaturated fat. Now, as you can imagine, if these are built within a triglyceride, there's going to be a lot more bending. Why is that relevant? Well, that's going to directly impact its melting point. Because now these chains are packed a lot more loosely, it means that less energy is needed to melt it. And unsurprisingly, they tend to be liquid at room temperature. Hence, we wouldn't call these fats, we call them oils. So that's where this comes from. Fats are effectively saturated fatty acids because they have a higher melting point, making them solid at room temperature, whereas oils are unsaturated fatty acids that tend to have a lower melting point and thus liquid at room temperature. Humans kind of also created a new kind of fatty acid, and that being unsaturated trans fatty acids. Now, again, trans in this case means different or on different sides. So they actually have a very similar structure to that of saturated fatty acids in that they're not going to show bending. They tend to have a fairly linear structure. So what would you predict their melting point is? It's higher. And that's precisely why they were invented in the first place. A process called artificial partial hydrogenation is where you take originally a polyunsaturated fat and you actually can convert it to a trans unsaturated fat. And the reason for this is, as you guys just predicted, it increases the melting point. Why would we care? Well, it's because fats can keep longer without spoiling. This was specifically done so that foods can last longer. Now, you probably know a lot of foods that have trans fats because they're typically categorized under processed foods. So a lot of processed foods, like pastries, like fries, donuts, things like that, if the source of the fat are trans fats, They sometimes are, again, colloquially known as processed foods or processed fats, and that's what these fats are. I think you can probably maybe find them naturally, but incredibly rare. These are almost all entirely man-made. Something that we'll look at a lot, and I'm just going to preview it now, we will eventually get into what are the dietary or health consequences of these fats in diet. The preview, the too long didn't read version of this, is that if possible, you should promote polyunsaturated fats in your diet, and you should minimize saturated and outright eliminate trans fats if you can in your diet. The reason that this is recommended is because of their relative risk factors for certain diseases. So saturated fats and trans fats have been strongly associated with cardiovascular disease. They have a fairly high risk factor for developing these diseases, whereas diets that have proportionally more unsaturated fats can actually lower the risk of cardiovascular disease. Another type of lipid that you've almost certainly heard of would be waxes. So waxes are another group of lipids that you've no doubt heard of because they're kind of in everyday life. And waxes are formed from the reaction between alcohol and fatty acids, and you get this ester group with these long hydrocarbon chains. And so as you can imagine, these pack together quite nicely, and they also have a fairly high melting point. So they're typically solid at room temperature. Now imagine all the waxes that you see in everyday life. Waxes on your lips to prevent water loss, or in nature, we've also explored this in A1.1 water, and we'll explore it even further in gas exchange in B3.1, but waxes can actually be used on the upper layer or the cuticle of leaves to prevent water loss. And this ensures that water is only passing through the stomata on the bottom of the leaves. Now, another type of lipid that we've already discussed in this class are steroids. So a steroid are lipids that are made up of 4 joined rings together. And it's actually 3 cyclohexanes and 1 cyclopentane. And some of them may even have a hydrocarbon chain. So a cholesterol is usually the progenitor or the originator of other types of steroids in the body. So we have cholesterol here, and now cholesterol as itself is actually used to regulate membrane fluidity, and we'll talk about this when we look at the structure of membranes in B2.1, but it's an integral part of mammalian membranes. And cholesterol can also be used to create specific hormones, stuff like testosterone or estradiol. And where these are really relevant is that these are the major male and female sex hormones, and they're involved in primary and secondary sex characteristics, things that we'll look at when we look at chemical signaling and even in reproduction. Now, what is the relevancy of these structures? Well, testosterone and estradiol, because they are nonpolar, they're able to directly pass through a phospholipid bilayer. And this is something that we've also explored in A1.1. So as a signaling molecule, which is what hormones are, the fact that they can readily pass into a cell means that they can deliver a signal very, very quickly. Now, the last type of lipid that we're going to look into is a very special lipid, and it's one that we're going to dive a lot more deeply into when we study membranes, and that is a phospholipid. So as we've already discussed, a phospholipid is composed of 3 different components: a phosphate group, a glycerol, and fatty acids, specifically 2 fatty acids. Now, these fatty acids may be composed of saturated or unsaturated fatty acids. So if you ever see a phospholipid with a bend, that bend is a cis-unsaturated bond in one of these hydrocarbons. Now, this molecule cannot be overstated in just how important it is for biology, and that is because it has a polar section and a non-polar section. So this phosphate group is actually polar, meaning that it is hydrophilic and able to form interactions with water, but these tails, or hydrocarbons, as we've already discussed, are non-polar and are hydrophobic, meaning that they will not interact with water. Now, because the molecule is both hydrophilic and hydrophobic, we can describe it as being amphipathic. And amphipathic means it is both hydrophilic and hydrophobic. Now, why is this relevant? Well, because if we place a phospholipid inside of water, they're naturally going to form these structures. If water is only on one certain surface, you can actually get a monolayer forming where all the phosphate heads are going to face the water and the tails will face away. And if you place a bunch of phospholipids in water, they will naturally interact to make a bilayer, where all the hydrophobic chains are going to face inwards and avoid the water, and all the phosphate polar heads are going to face towards the water. So you form this hydrophobic core in this hydrophilic exterior, both facing the extracellular and intracellular space. So overall, I would say lipids are quite variable and quite different from other kinds of biomolecules, because whereas the other biomolecules are really the same monomer being linked over and over and over again, this is not necessarily true for lipids. They may be composed of a variable amount of components, such as in the case of triglycerides or phospholipids. What is the use of lipids in animals, i.e., where are they stored and what kind of different functions do they have in certain organisms? So first of all, where do we store fat or triglycerides? So triglycerides typically are stored under the skin in a separate tissue called subcutaneous fat. So sub means below, and cutaneous refers to the skin, so it's below the skin fat. And if I were to zoom into here, here is an example of what this might look like in an Arctic or a aquatic animal where heat conservation is a really big priority. And notice that this layer of fat here is really, really thick. In these organisms, we call this specific thick layer blubber, but really it's just a bunch of these cells. So these cells would be called adipocytes, or adipose for lipids or fats, and cyte meaning cells. So they're literally fat cells. And these cells, as you can see, are effectively large storage vesicles for triglycerides. So what are some uses for triglycerides? Well, for one, something that we've explored when we looked at water, they're a really good buoyancy aid, meaning that the more of these triglycerides or lipids that are found in an organism, the more buoyant they will be in water. Well, why is that? Well, as a review, if the object's density is greater than the fluid, then it's going to sink. If the object's density is less than or equal to the fluid, then it's going to float. So what's fat's relevancy in this? Well, if we compare, let's say 2 kilograms of fat versus 2 kilograms of muscle, these have the same mass, but notice their relative density. Fat is significantly less dense than, let's say, muscles are. And so for the same amount of each, fat is going to take up more space, thus be less dense. And as a result, if I want to increase buoyancy, I'm going to increase the amount of fat that I have, making it much, much easier to float inside of water. And we see this as an example in things like seals, where seals can have upwards of 40% of their mass is actually blubber. So it allows them to float very, very easily while expending very little energy. Another use that we've also explored in A1.1 water is fat is a really good thermal insulator. So a heat conductor means a material in which heat can easily flow through, something like, let's say, a metal. Whereas a heat insulator or a thermal insulator is one where heat or energy does not flow through easily. And so fat is an extremely good thermal insulator. And so for things like endotherms, like us, or warm-blooded animals, they are trying to maintain a constant internal body temperature, which means that our body temperature, or at least my body temperature right now, is significantly hotter than my surroundings, which means I am constantly radiating heat. I want to try to hold on to that heat as much as I can, which means that the more and more fat that I have, the more of that heat is actually being trapped. So if I'm an animal, let's say, that lives in the Arctic, something like a polar bear or again a seal, I can have really high proportions of fat, and that's going to act effectively as a heat trap. So all that heat that I'm radiating in my body, more of it is going to be trapped because of all of the fat. So less of it is going to be radiated to the atmosphere, allowing me to stay warmer. This operates in the same way that clothes make you warm, right? Clothes don't actually provide me any heat. They don't provide me any energy. What they're doing is they're acting as a barrier to the heat that my body is radiating. The more clothes layers that I add, the more of that heat that I'm radiating is trapped. And that's effectively how this works. Another advantage of fats is they act as a means of protection. And it may be something that you've not even be aware of. So in us, we don't just store fat underneath our skin in subcutaneous fat. We can also store it around our organs in something called visceral fat. So we can actually use it around our heart, or even around our gut, and that acts as a means of protection and as a way of shielding from damage, because they can actually kind of act as a cushion. And this is especially relevant if you really look actually at the soles of your feet, where you actually have these fat pads, and that actually it acts as an absorbent to the really high amounts of shock that would happen every single time your feet strike the ground. So on these little soles of your feet, so to speak, and on your toes, you have little stores of fat that actually act as a cushion. And finally, probably the most relevant focus of this subtopic is fat's use as a long-term energy storage molecule. So what about it makes it really good at a long-term energy storage? So let's first of all discuss what is the difference between short-term energy storage and long-term energy storage. Short-term energy storage is energy that I need to access very, very quickly, and energy that I can store very, very quickly. So an analogy here would be like cash in my wallet. If I need to add money to my wallet, I can do so by literally opening my wallet and putting some money in. If I need to spend money, I can literally take money out and spend the cash very, very easily. So any cash that I have in my wallet on hand is cash that I'm going to be using all the time very, very easily. However, it's not economical or even advisable for me to have my entire net worth in my wallet. It would be very cumbersome and not very efficient. So the majority of my wealth would actually be stored long-term in, let's say, a bank. And that's kind of what long-term energy storage is. I can store a lot more of it more reliably in, let's say, my bank account, but accessing it is slightly more tricky. I have to maybe go to an ATM or a teller, and I have to do extra steps in order to access it. Same thing if I want to actually add money to that account. There has to be certain extra steps that didn't need to occur when I placed cash in a wallet. So what about lipids make them really good at long-term energy storage? Well, first of all, in terms of efficiency, gram for gram, they release twice as much energy as carbohydrates when we oxidize them in cellular respiration, which is phenomenal. And we're going to look at this more when we look into cell respiration in C1.2. But it means that if I have 2 grams of a lipid or 2 grams of a carbohydrate, I can get nearly twice as much ATP out of that lipid than I could out of that carbohydrate, making it really, really effective for getting energy when I need it in a really dire situation. Another advantage of lipids is that if I'm going to hold something long term, I want to hold it in a very efficient manner. And lipids, as we've just discussed, are hydrophobic. So that means that when I store lipids, I can store them as pure lipids, no water around as well. That's not true for carbohydrates. If I were to store the same gram of carbohydrate as I would lipid, I would also need to store a bunch of water along with it, because water is going to naturally be attracted to carbohydrates. So it becomes a lot less efficient to store huge amounts of carbohydrates, because you would have to also store all that water along with it. So storing lipids is much more efficient because I can store pure lipids without any of that water. And in fact, this is what you see. So animals that are not going to have ready access to eating or to acquiring nutrition very regularly need stores of fat that they can access, let's say, on travel. A great example of this are migratory birds. Here's actually a migratory bird that I studied in my master's called a snow bunting, and they migrate from the very bottom of Canada all the way to the Arctic. And that's a very, very, very large distance, and they don't have much time to actually feed along that travel. So they do a lot of their kind of energy consumption on the wings, so to speak. So before their migration, they actually put a ton of fat stores right around their chest, and they can actually access this incrementally while they're flying, which is a very high energy intensive process. And because they can store this as pure fat, it actually doesn't add or contribute that much to their mass, allowing them to fly still efficiently while acquiring energy as they go. Camels are a very famous desert animal adapted to very, very low water environments. And how exactly can camels survive this? Well, the misconception is that their humps are full of water. In reality, their humps aren't really full of water, they're full of fat. But what's really cool is that misconception wasn't entirely untrue, because not only can this fat be used as an energy storage, but it can be used to generate water. So when I'm breaking this fat down, not only can I access energy reserves, but the oxygen from the air can combine with all of these hydrogens in the hydrocarbon chain, and you can actually produce metabolic water. And actually, through cellular respiration, water is a byproduct. And so actually breaking down lipids provides more metabolic water than carbohydrates do. So it's kind of cool, because these humps, while yes, they're large stores of fat, they can be used for the organism to actually produce water in themselves as they're breaking them down through cellular respiration. It's kind of remarkable. So overall, lipids are kind of amazing because they have huge amounts of functions inside of the body. They can be used as a buoyancy aid, as a thermal insulator, as an energy storage molecule, and even as a means of protection. Something that's not here, but I'm just going to allude to as well, is that fat isn't only a good thermal insulator. Something that we'll look at in neural signaling is that fats are also a really good electrical insulator. So a lot of your nerves, actually, something like your motor neurons, those neurons that are sending signals to your muscles, a lot of them are actually coated in fat, or what it's called myelin, and that actually insulates the electrical signal and it actually increases the speed of electrical transmission. So fats are also not only a good thermal insulator, they're a good electrical insulator as well. The last part that we're going to talk about here is we've looked so far at carbohydrates, we looked at nucleic acids, we've briefly looked at proteins, and we've now looked at lipids. Is there such a thing as combining some of these things together? Yes. So biomolecules can exist in combinations. Here's one combination, a glycoprotein. And again, where does that name come from? Glyce, or glyco, usually refers to sugar, and well, protein. So it's literally a sugar combined to a protein. So in this case, what I've shown here is here's a membrane, here's my phospholipid bilayer. In red, I've represented a protein, and green here is a polysaccharide. And because this polysaccharide can be multiple, there's no definite size or shape. Sometimes this is referred to as an oligosaccharide. These glycoproteins are embedded within the cell membranes. A similar example to this would be a glycolipid, where you're combining, again, an oligosaccharide with a lipid, which is also embedded in the cell membrane. And then finally, another combination that we've already explored would be a lipoprotein. We said that if we want to transport something through the bloodstream that is non-polar, it's not going to be able to dissolve, so how do we move it? Well, we package it inside of a lipoprotein. Take a look at this structure. How does this structure work? How does this structure allow the body to transport hydrophobic molecules in a hydrophilic solvent? So the hydrophilic heads and these sections of the proteins, these are forming hydrogen bonds with water. That's what's allowing that thing to dissolve. But the interior, see, it's a monolayer here. All of these hydrophobic parts, that's what's interacting with these hydrophobic packages. So it's almost like I want to package something that itself can't dissolve. I'm going to put it in a capsule almost. And the exterior of that capsule is hydrophilic, but the interior is hydrophobic. So that's how I can transport these molecules across the bloodstream. Let's take a look at these two sections here. Both glycoproteins and glycolipids are embedded in the plasma membrane, but they're embedded in a very particular way. They always will face the polysaccharide portion, will always face the extracellular space. That means that if I have a cell, all of the glycoproteins will face in that orientation. They will all face outwards from the cell. Now, what is the significance of that? Well, that has all to do with the main role that they have, which is cell-to-cell recognition. So glycoproteins' major function within cells is a way for one cell to communicate with another cell and one cell to identify self from non-self. So it's maybe a question that you've never considered. How does your cell know what should be in your body versus what shouldn't be in your body, i.e. foreign? How does it know this? Well, glycoproteins play a huge role here. Your body is able to communicate and identify self from non-self by being able to bind to these glycoproteins. And this is a concept that we'll look at when we do defense against disease. But essentially, these glycoproteins act at what are called antigens, or basically surface markers. And your body cells have receptors that can interact with these antigens. And if they identify this antigen as, well, this should not be here, that would initiate an immune response. And that will cause that cell to get destroyed. Versus if they identify the cell and they're like, oh, yep, this is fine, it'll leave it alone, so to speak. So these glycoproteins they have a lot of functions, but their major function that we're exploring is in cell-to-cell recognition. And the example that we're going to use to study this is red blood cells, or your erythrocytes. So your erythrocytes, so the true name for red blood cells are erythrocytes, and that's just Greek for red cell. That's literally what that means. Cyte is cell, and erythro here is red. So your erythrocytes are just like any other cell in your body. They also have surface markers. So they have glycoproteins on their cell membranes that are all facing out. Now, in the human system, we have three major kinds of glycoproteins. We have the O, the A, and the B. Now, this is extra. You do not need to know this, okay? This is just more if you're curious as to the structure of these. This is the true structure. Notice that the differences are very, very subtle. Now, if you also notice a pattern here, look at the base structure of the O antigen versus the A and B. The A and B have the exact same portion of that O. The only difference is that they have an extra different carbohydrate on top. So this is why you'll normally see something like O has no antigens, A has A, and B has B. But O does have glycoproteins, but they're effectively the same base as both A and B. So just keep that in the back of your mind. We'll be looking at this when we do genetics more. It's not a learning outcome yet that you understand the genetic transmissibility of these blood groups. We will be exploring this when we do genetics, but I wanted to include it here just as an aside. Now, let's explore how different combinations allow for blood donations and blood recipients and how this whole system works. If you are blood type O, then that implies that you have O antigens, but more importantly, it means that you lack A and B glycoproteins. So you have no A glycoproteins, you have no B glycoproteins on the surface. If you are blood type A, then that means that you have only A glycoproteins on the surface. If you are blood type B, that means you have only B glycoproteins on the surface. And then finally, if you are blood type AB, it means you have both A and B glycoproteins on the surface. Now, if I am blood type O, then the kind of antibodies that I'm going to produce would be, I want to look for any A glycoproteins, and I want to look for any B glycoproteins, because both of these would be considered foreign. So the question is, what blood groups can someone with type O donate to, and which blood groups can someone with type O receive from? So O is sometimes referred to as the universal donor because it doesn't have any antigens that any other group would identify as foreign, right? Remember, the O antigen is basically the base of the A and the B glycoprotein. So it can donate to O, it can donate to A, it can donate to B, it can donate to AB. However, it can only receive from O. If it received from any other blood group, if let's say someone with type A wanted to donate, it will produce antibodies that will identify that as foreign and destroy it. The same thing with B, the same thing with AB. Now let's look at the converse scenario. If an individual is type AB, who can they donate to? Who can they receive from? Type AB is known as the universal recipient because anyone can donate to type AB because nothing will be deemed as foreign. If someone with type O donates, fine. If someone with A donates, fine. If someone with B donates, fine. Nothing is deemed as foreign. However, they can only donate to AB individuals. Because if they donate to A or B or O, something is going to be identified as foreign. Now, it should be noted, and as I mentioned in my notes here, that this is a simplification because in real life there's actually 8 blood groups because there's an additional antigen called the Rh factor that is taken into account. If you don't need to know that in this course, but if you have that extra Rh factor, then you are positive. If you lack that Rh antigen, then you are negative. So the true universal donor is which blood group, and the true universal recipient taking Rh into account is what? O negative is the universal donor, and AB positive is the universal recipient. But if you ever end up donating blood, which I also encourage you to do, they'll usually type you the first time you donate blood as well so that they have your blood type on record. So overall, this section really is just about what are glycoproteins and what is their role in cell-to-cell recognition using the ABO system as an example. Okay, let's go over the learning outcomes for subtopic B1.1 Carbohydrates and Lipids. And note that there are only standard level learning outcomes for this subtopic. Now, the very first learning outcome that we explored was what makes carbon as an atom so useful as the base of all biomolecules. Why is it that all of life is, quote, carbon-based? What properties does carbon have that allow such diversity to take place? We also then explored how different SI units can be converted between using some simple math calculations. And then we kind of summarized how all biomolecules, proteins, carbohydrates, nucleic acids, and lipids can be built using condensation reactions and can be broken down using hydrolysis reactions. And these were explored in the following learning outcomes. This learning outcome, this learning outcome, this learning outcome, and this learning outcome. All of these learning outcomes effectively explored how these biomolecules can be built or broken down using hydrolysis and condensation reactions. And then kind of the rest of these learning outcomes is broken up broadly into carbohydrates and lipids. This had all to do with carbohydrates. This had all to do with lipids. So with respect to carbohydrates, we first looked at the form and function of monosaccharides, or the building block of all sugars, and that led into polysaccharides and what makes them such good energy storage molecules. And we mainly explored that for glycogen, amylose, and amylopectin. And then the fourth polysaccharide that we explored was that of cellulose. And this polysaccharide is not used as an energy storage molecule, but rather as a structural component of plant cell walls. The last learning outcome we explored here was that of glycoproteins and their role in cell-to-cell recognition that allows cells to recognize self from non-self and potentially initiate an immune response. With respect to lipids, we looked into what makes lipids hydrophobic, what about their properties cause all lipids realistically to be hydrophobic and not miscible in water. And then we looked into the differences between fatty acids, that being saturated, monounsaturated, and polyunsaturated, and what differences these have with respect to their properties and their functions in living organisms. We then looked into triglycerides and their roles as both long-term energy storage molecules and with respect to other properties such as thermal insulation. And then we looked into how phospholipids are a consequence really of their amphipathic nature, or the fact that they're both hydrophobic and hydrophilic. And then finally, that nonpolar steroids, because they're able to pass directly through membranes, some of these can actually act as really good hormones, such as testosterone or estradiol. If you wish to learn more, this is really a good spot to start. These are books in which I use to help build my notes and the relevant page numbers if you wanted to read further. And what I think is especially relevant in this topic is exploring certain biomolecules in the 3D space. And I've linked a bunch of 3D molecules that really allow a better understanding as to the differences between these different forms, between polysaccharides and monosaccharides and of various lipids. And I think I can only do so much by teaching something in a 2D space, which is why I always recommend looking at them in a 3D space. And then the article I listed here is something that's really, really interesting, and it has something to do with glycoproteins. And that is, there was a fairly recent paper and breakthrough discussing the potential use of artificial blood that would eliminate the need for certain donations or certain recipients with respect to blood groups. So this is an extremely interesting paper to have a look at. And lastly, I would always encourage students not to learn just for the sake of a test, but learn for the sake of learning, learn for enjoyment.